Hello, everyone. This is Felix Dunn with AcCELLerated Biologics. For those of you that are on right now, welcome. Thanks for attending. We have a pretty good, concise little webinar that we're happy to share with everyone tonight. We're going to start here shortly. Scheduled time is 6:45, and we're going to give everyone a few minutes to log on in case they have a few timing issues. But I wanted to let everybody know before we get started that if you've used GoToWebinar before, there's a couple of ways we'd like for you to ask questions when the time has come. For the first part of the webinar, we're going to have some speakers from ForeverLabs and Databiologics. And then after that, we'll be doing the town hall question and answer session. So if you want to raise your hand and ask a question live, under the attendees tab, there's a little mark there where you can raise your hand, and that'll signify to us that we can call on you and you can ask your question to the whole group. And then also under the questions tab, you can write your question if you don't need to have a discussion going on about your question. So just want to let everybody aware of that. And I'll make sure to remind anybody if they have questions that they can raise their hand and or write out the question in part of the question display on the dashboard of the GoToWebinar page here. So we are here at 6:45. I'm just going to give it a few more minutes, 2 more minutes, and then we'll get going. Again, this is Felix Dunn with AcCELLerated Biologics. We're going to get started on our SuperShot technology town hall meeting. To get started, I'm going to give the platform over to Stephen Klausnitzer, the CEO of ForeverLabs, and let him get this webinar started. So Stephen, go ahead.
Thank you, Felix. Thank you everyone for joining and attending. I'll try to keep this really, really short. So thank you to AcCELLerated Biologics for setting this up. We are working with some of the greatest physicians in the orthobiologic regenerative medicine space across the country because of AcCELLerated Biologics. So thank you, and thank you for continuing to host these events. So we started ForeverLabs back in 2015, and our goal at ForeverLabs is to leverage autologous biology in regenerative medicine. So we're a regenerative medicine company that works with autologous biology. I'll let Mark and Anne, my colleagues, talk more about the product at hand today, but just know that that is our mission statement. We are a regenerative medicine company, and we use autologous treatments. We made a declaration at ForeverLabs when we started the company back in 2015 that we would never do anything outside of the good graces of solid science and medicine or the FDA. So just know that we check all of those boxes, and we can talk more about that as we go through this. And I think you'll just get a sense of that as you hear from Dr. Navity, my co-founder, Mark Karakowski, and our COO, Anne. I also want to say that I think it would be really easy for us to launch this product broadly without collecting a lot of data, but that's not how we go to market as a company. We're working really closely with Databiologics, and Dr. Bowen's going to talk about that a bit, to gather data. We want to do the Pepsi challenge and show that SuperShot PRP is working, is more efficacious than standard PRP, and we're confident that it is. And Dr. Navity can talk about the results that he's already seeing in that regard. So with that, I'm around for any questions afterwards. Just want to say thank you to everyone for joining. We're really proud of the product that we've created, but we're most proud of the caliber of physicians that are using it. I mean, it just says a lot about what we've built. So thank you for being here. With that, I'll turn it back over to you, Felix. Thank you.
Great. Thank you, Stephen. So next on the list for presenting tonight is Mark Karakowski, Chief Science Officer at ForeverLabs. He'll go into the nuts and bolts of SuperShot and its development. And again, for any of the attendees, if you have questions specific to Mark or Stephen or anybody on the panel, during any part of this webinar, you can type in your question on the question tab on the dashboard. And then when we get to the open forum after all the presentations, you will be able to raise your hand when you see the little hand signal by your name in the attendee list. So a little more just upkeep information. And now, Mark, off to you. Go ahead.
Hi, everyone. Thanks so much, Felix, and thanks for AcCELLerated for putting this together. My name is Mark Dudakowski. I'm the President and Chief Science Officer of ForeverLabs. And as Felix mentioned, I'm going to getting a little bit feedback. Did somebody just turn on a mic? Try. Yeah, now I think it's. Oh well, I'm just hearing myself echo. I don't know if somebody.
Maybe if you're not talking on the call right now, if you could go on mute, that would be very helpful. Thank you.
Yeah, that's better. Thanks, whoever. So, yeah, my background actually. So I'm a PhD, and my background is in medical physics. And starting in 2001, I was a researcher at Henry Ford Hospital in Detroit, Michigan, looking at mesenchymal stem cells and bone mesenchymal stem cells, and using them to treat stroke and other neural injury. And then that kind of grew, and we found that these cells had this great therapeutic value, and we found somehow how they kind of mediated this therapeutic value. And in 2015, as Steve mentioned, we formed ForeverLabs together. And through ForeverLabs, I became aware of OrthoBiologics and bone marrow concentrate therapy and PRP therapy. And based on what I had learned in the mesenchymal stem cell research space, over the 15 years prior, I saw the PRP system, and it made all sense. It made a lot of sense to me that yeah, there is therapeutic, molecules of fraction of the blood that could be leveraged for healing and especially to kind of condense the inflammation period and extend the regeneration after injury. But what struck me was that the platelet poor plasma fraction was quite large and just tossed after the spin when you prepare your PRP. And from what I knew from my mesenchymal stem cell day research is that the plasma is full of extracellular vesicles, and these are extremely therapeutic molecules. They're little lipid encased, almost acellular packets that contain a lot of proteins, but then also non-coding RNAs that actually can directly regulate gene expression. And that's what we found. That was one of the large mediators of the therapeutic value of the mesenchymal stem cell, was these extracellular vesicles they produce. And so this is something that's already in PRP, but from the physics of it, when you're spinning whole blood at, say, 4,200 RPMs on a tabletop centrifuge, you're not concentrating that fraction. And so I strongly believe that it's a therapeutic fraction of the PRP, but that it's not being concentrated in the standard PRP prep. And so that kind of dawned on me. I said, Steve, I think we should look at this. And so I didn't invent a new physics, but I did apply a known physics to this problem. So how can you at a tabletop centrifuge speed concentrate these therapeutic extracellular vesicles? Well, in our lab, we used to use like a washing machine size centrifuge that spun 100 times faster and overnight for 24 hours. But there is kind of a trick of physics that you can use to get these low density lipids out, and it's called a two-phase aqueous separation. And so if you add to the plasma just a little of polyethylene glycol, a little bit of a high weight sugar, a high weight dextran, what you'll get is they'll both solubilize in the plasma, but each of these soluble phases, and they'll separate really easily. It's just like oil and water when you spin them at a low centrifuge speed. But both of these phases, the one that contains the upper one will have the PEG, and the lower will have dextran. These low density lipids, these extracellular vesicles, are not soluble in either of them, and so they'll be actually expelled. They're immiscible, and they'll be expelled at a very low speed. And so you can use this kind of trick of physics, so their size excluded, and they'll spin out. And so what you can do, and what we created with this process we call the SuperShot, is you can do one more spin of that platelet poor plasma. It takes 1 minute, you spin it 4,200, and you'll get a precipitation, you'll get a pellet of extracellular vesicles. And what are these extracellular vesicles? Well, as I said, they're already in the plasma. It's already in your PRP. They're just not being concentrated. And Steve's got it. So what you've got is a variety. There's many different species. Like, it's known that exosomes is one of them, but we looked at them with a nanocyte. So we looked at them individually with a laser, and you can size them, and they range anywhere from 30 nanometers to 450 nanometers, and that can include exosomes like the autologous, but that also includes platelet microvesicles, other microvesicles, just different. There's really a lot of species of these EVs. We call them extracellular vesicles, or EVs, in the blood. We analyze them using like I said before, one reason why they're so therapeutically interesting is because they contain a lot of these microRNAs, these short non-coding RNAs that directly regulate genes. And so we did some assays to that effect and found that there's hundreds of microRNA species in them, and they're full of them. It's just a very rich in microRNAs. I believe it's, probably one of the therapeutic fractions of PRP as is, just not being concentrated. So what we've done is developed a tool to allow you to concentrate that fraction. And in doing so, there is preclinical evidence that plasma extracellular vesicles, exosomes included, have therapeutic value. Preclinical evidence where they've shown that it's improved wound healing, improved bone healing, and so it is known that these are therapeutic. So it stands to reason that if they can be concentrated in PRP, you may have a more therapeutic. But that's what we want to find out. That's why we're returning to the docs. That's why we're returning to Databiologics, and we're interested in doing that challenge as Steve talked about. And so what we really believe we've got here is perhaps a higher fidelity PRP. There's another fraction that you can adjust for therapeutic ends, and we think that the science really does stand behind the fact that this fraction is of interest and should be part of a PRP prep if you can. And so that's what we provided. And following me, which I'll pass on, is to our Chief Medical Officer, Dr. Navity, who's actually been using SuperShot, I think the longest as a physician, and so he can talk a little bit about what he's learned in the way. But yeah, I'll be here at the end, happy to answer any questions. It's hard to talk about all of that in a short 10 minutes, but thanks for your time, and happy to talk to you, answer some questions at the end. And I'll pass it on here to Dr. Navity, Dr. Tom Navity.
Great, so we're gonna get Dr. Navity to speak next. And while I bring up his presentation, just let everybody know that, like Mark said, he's probably done more SuperShot PRPs than anyone. He's put together a little case study for you guys to review today. He is board certified in both physical medicine and rehabilitation and pain medicine. And as we get this lined up.
Thanks, Felix. Hi, everyone. I'm Tom Navity, Dr. Navity, and as Mark alluded to, I started using SuperShot almost a year ago. I just looked up. My first patient was treated December 4th of last year. So we're closing in on 1 year. I've been using it exclusively with my PRP and put together just a very small case study with comparing two patients. I have been, and Dr. Bowen will talk about next, I have been using Databiologics now for close to 2 years. And so I had some data on the patients that I had treated with PRP prior to incorporating SuperShot, and then obviously have data after incorporating SuperShot starting in December of last year. And as we can see in the case presentation we'll look at, similar type patients in this study here, both female patients in their 50s with rotator cuff disease, partial thickness on MRI testing, different baseline quick dash functional scores, but you'll see the reduction in score actually even favors the SuperShot PRP patient more. So basically what I'm trying to say here is they're very similar patients in their demographics and their severity of illness. And then they're non-smokers. If we advance to the next slide, you can look at the difference in the composition of their PRP. So in our lab, we quantify all of our PRP, which I personally think is a really important step, knowing essentially the dose of PRP we're using. I think there's been plenty of good papers and presentations with the end result or the conclusion being not all PRP is the same. So if you're not quantifying what's in your PRP, you don't really know. And in particular, in this version of PRP, very similar in our platelet concentrations, in our white blood cell concentrations, and our red blood cell concentration. So the only difference in the quality or the type of PRP was patient A did not have SuperShot added back, whereas patient B did undergo the SuperShot process and had the extracellular vesicles added back to the PRP. Both injections were given with ultrasound guidance. into the supraspinatus tear. Both received a 3 cc injection into their supraspinatus tear. So again, I like this this comparison because it's essentially the same patient. It's essentially the same procedure, with the only variable being whether SuperShot EVs were added back to the PRP or not. So if you look at the outcome data that we tracked through Databiologics, very similar 3-month outcomes, very different 6-month outcomes. So the functional scores were higher for patient A than patient B, and then the gains were obviously much greater for patient B. It's an N of 1. It's a very small study. It's not sufficient to make any large conclusions, but this is an example, and it does highlight what I've seen clinically. And I guess what I've seen clinically in using this, my follow-ups I do at 1 month and 3 months. And at my 1 month follow-up, I'm noticing much faster reductions in pain and greater functional improvements. And then at 3 months, those functional improvements are seem to be even greater, and the pain reduction is sustained. So in tracking these outcomes through Databiologics, hopefully we'll have a much larger sample size where we can make statistically significant conclusions whether or not adding the extracellular vesicles back to a very high quality PRP leads to greater functional improvement and reduced pain. That's the gist of my studies here, or my presentation here. Wanted to keep it very short, very sweet. But again, take-home points I think are we need to track our outcomes, and we need to document what we're injecting, what's in our PRP, and then also whether or not we've added back extracellular vesicles, created a super PRP, an enhanced PRP, and whether that leads to improved outcomes or not.
Great. All right. Thank you, Dr. Navity, for your information here. Now to kind of continue with the experience discussions, I'm going to bring on a few speakers that have also used the SuperShot. Dr. Paul Tortland, I see that you're here, and that if you want to go now, that would be good, or if you want to go later.
Whichever you prefer, whatever easiest for you.
Dr. Tortland, you have the floor. Go ahead and give us 3 to 5 minutes of kind of your experiences to date.
So we were one of the early adopters of the SuperShot PRP, and I've had discussions online and personally, both with Dr. Navity and with Mark Karakowski. We've been very, very pleased with the results. Primarily what we've seen is a decrease in the amount of inflammatory response to the PRP, and an increase in the subjective clinical outcomes. Now, we don't have enough data collected yet in terms of hard research to say that on an empirical basis, so it's purely subjective. But I was using exosomes long before the SuperShot PRP came on the scene, and for me it was like adding rocket fuel to the PRP. And there were certain circumstances where I actually would not even do the treatment if we didn't include the exosomes. And for me, that's particularly true for PRP for hair restoration and PRP for cosmetic applications. The SuperShot PRP is an absolute home run for those particular applications. Just because of the added expense, I don't use it for every PRP. I try to maintain some cost effectiveness for patients. But for those patients who I think could benefit from an added boost, and for example, somebody who has more advanced OA and an autologous stem cell treatment such as bone marrow or adipose may be out of their price range, but I don't think regular PRP would be quite strong enough to get the job done. I think the SuperShot PRP for us has been an absolute boon for those patients. I recently treated a patient just the other day, 72-year-old woman who looks like she's about 55, but we did a rotator cuff injection and a hip injection, and this was the first time we had used the SuperShot on her. I've treated her extensively in the past, And she called back within a few days, just absolutely raving about how much better she felt and how there was no inflammation following her treatments. And this is somebody that I've done a lot of PRP on over the years. So there's clearly something going on with the addition of the exosomes and other growth factors that are extracted through the SuperShot PRP process that are having some type of impact. We do have a cell counter in our office, and we measure all of our PRPs, so we know exactly what we're injecting. And we're hoping just to collect the data. We're part of the Databiologics Consortium, of which Dr. Jay Bowen is involved. We were one of the early adopters of that also. So we're hoping that over time, as we send the data in, we'll start to get more hard data to ideally back up what we're seeing experientially.
Great. Thank you very much, Dr. Cortland. And again, for any of the attendees that have a question for Dr. Cortland, Mark Karakowski, Dr. Navity, you can write your question down in the question section on the dashboard, or when we get to the open forum, I will be able to unmute you if you raise your hand in the attendee section and let me know that you want to ask a question. And at this point, I want to get to another physician that has used this product. And Dr. Altizer, I see that you are on the board here. I've unmuted your microphone. Can you hear me, sir?
Yes, can you hear me?
Yep, we can hear you loud and clear, doctor. So Dr. Altizer, he is board certified internist, specializes in physical rehabilitation and regenerative medicine, has a great practice in Charlotte, North Carolina. And Dr. Altizer, if you could share some of your experiences with the group, that'd be great.
Well, I don't have a lot of experience with Super PRP yet. I've done 22 patients, 5 knees, 3 low backs, 6 shoulders, a couple of elbows, and a couple of Achilles tendons, and a couple of wrists and thumbs. All of them have done well except the thumb and the wrist. Don't know why they didn't respond well, but all of the other patients have had a really great response and have been very happy with their treatment. The question I have about Super PRP is, as far as the making of the Super PRP, we only use 4.5 cc's of the platelet poor plasma to mix it up. And my question is, why not use a lot more? If we're trying to get exosomes, isn't more better than less? And so why don't we use a lot more plasma to get even more exosomes? That's my question.
Mark, you want to take that one?
Yeah, I'm
Yeah.
happy to. Or Tom, did you want to jump in?
I could definitely comment on what I do, and I'll let you talk first. Science.
Oh, okay, sure. First of all, yeah, it's a really good question, and yeah, is more better? Probably is my best guess. What we do know from our in vitro work, and when we're treating cells we've actually treated cultured mesenchymal stem cells from both bone, marrow, and fat with these cells, and with the extracellular, the EV fraction from the platelet poor plasma prepared by SuperShot. And so that when we do that, those cells grow much more readily. And we actually have some evidence too that there is a delayed kind of artificial aging that happens in vitro. When you divide cells quickly, they start to have this go senescent more quickly, and that's delayed. And we have seen in those experiments a dose response curve, and it does stand to reason that what we're suggesting with the kind of the standard SOP for SuperShot PRP is that you're doubling your EV concentration. So you're taking whatever your volume of PRP is, you're using about that much platelet poor, and you're getting the EVs out of that, and so you have doubled the EVs in your PRP. I want to say that it's very likely that that's not at the top of the curve, and that you could go higher. It's the place where when we developed the SOP, we felt that we had double would create a significant effect. Would be significantly different than not doubling it, and so that would be measurable and yet conservative. And but they're just scientifically, that would, speaking, that's probably not near the saturation point. You can in an in vitro situation, you can saturate cells with too many extracellular vesicles or exosomes, but it's a really high concentration. And I think under most therapeutic applications, the volumes you're talking are so large that that's probably not going to happen with the PPP you have to work with. So that's really up for with you physicians and data biologics to answer for us. I think in the end, what we've, like, I'm a research scientist, and we created a tool, and so we started at what we thought is a very I guess reasonable and conservative starting place. But if this tool says, that you get led to say, hey, we need to move a little bit higher, maybe even when it's joints, I don't know. That's up to, that's my 2 cents, and kind of from the scientific standpoint.
Yeah, so I'm clinically, I'll just kind of cut to the chase. I'm using 12 cc's now. What I want to do is, and what we're working on is being able to quantify how many EVs are in the sample, so that we can study it, we can track it, we can see where that bell curve exactly is. But until we have the ability to quantify them, we won't really know. And I would say just kind of use your clinical judgment, and like we do with most things. And I have not noticed anything bad happening by using more platelet-poor plasma. I can't say I've noticed even better results, but I definitely see a bigger pellet at the bottom. So I know I'm getting more EVs, and would like to start quantifying those and tracking those. And one of the wonderful things that Databiologics has done is one of the questions when you enter in whether you use SuperShot PRP or not is how much PPP was used. So at least from that standpoint, we can try to find a optimal amount of PPP that's going to be used just by looking back at the data and finding outliers or finding the best outcomes, and then seeing how much PPP was used to create SuperShot in those patients.
Great. Thank you, Dr. Navity. Next, I'm going to bring on Dr. Gislaine Robert. She has practiced out in Redmond, Washington, board certified sports medicine physician. And Dr. Robert was seeing a patient, but she recently joined us. Dr. Robert, can you hear me? I see, Dr. Robert, that you did type in a question. Is it accepted in Canada? We will get to answer that, but if we don't hear you right now, I'm going to put you back on mute, and you can ask a question to me when you're back on and ready to go. But to kind of keep the webinar moving, I'm going to bring in Dr. Jay Bowen, who's been mentioned here a couple times. He is with New Jersey Regenerative Institute and Data Biologics, and he is board certified physician, specializing again in physical medicine and rehabilitation, sports and pain medicine. And he is going to now kind of discuss Data Biologics and its role with this whole program. So Dr. Bowen, the floor is yours.
Thank you. And I want to extend a large gratitude to AcCELLerated Biologics, ForeverLabs, and especially, as Dr. Torlin said, our early adopters of our platform that have helped us to build it and to make it something robust where we can start, getting this data about the questions that people are asking or thinking about. So again, just a big thanks for everybody, tolerating our growth as a new company, and we will be getting some good data. So I'm going to talk a little bit about, data. And so why do we collect data? Well, we have newer treatments. Let's see how they work. Let's see how they compare to the standard of care. Are they safe as a standard of care? Or are they safer? Are they as efficacious or not? How do they fit in the whole paradigm of treatment? Would you do, as Dr. Torlin said, a PRP on someone with knee OA, grade 1-2, that would get 1 year of benefit? Or if you add SuperShot, now they get 4 years of benefit, and they don't need an adipose procedure or bone marrow procedure. That's something we're going to try to look toward. Where does it fit in this whole continuum? Because we have lots of tools in our tool belt, but where do they all fit? And unfortunately, right now, medicine is dictated by insurance companies' reimbursement. So they might be a interested party to find out, with us collecting data, what's important. Our patients, probably really the reason we do it is we're doing it for our patients. When a patient comes in and says, hey, I read about this exosome thing, and they ask me, I go, I don't know. It costs a lot of money, comes from someone else, may have disease transmission. I have no data. Would you rather have your own stuff? So again, an informed decision about costs, outcomes, and adverse events are going to be important. And some people do it altruistically to add to the literature also. Next slide, please. So we're all taught in med school that randomized, placebo-controlled, level 1 studies are the best, the gold standard. But is that really the way it should be? Next slide, please. So, in the past, we looked at the world and we had a scientific method and we said, we observed things, we had hypothesis, we did experiments, and then we modified things. So, in surgery, it's really difficult to get a, control. In procedures, a similar kind of thing happens. It's difficult to pair up people and to do that, good control. So next slide. So to keep the thinking is, well, why, do we do research at all? Well, we want research to direct our daily patient care, to inform those patients. And the best way probably to do that is real world data. But, we started doing this in 1995 or so with pen and paper, and it was very onerous. I was the fellow, and I got to do all the grunt work. So that was not so fun. And back then, we didn't all have smartphones, and we didn't have the technology to do actually what Databiologics is trying to do now. So, we want real world data with large data sets that's easily accessible, researchable, and relatable and practical. And it's probably likely the reason, from the randomized placebo-controlled trials, why we don't have the answers we want. And so we have a bunch of experts that make these expert consensus, these guidelines, these review articles. And we sometimes have more meta-analysis than we actually have original research and published in a year. And so, we have these guys who sit in an ivory white tower who make up guidelines that are onerous to us physicians on a practical base, seeing people daily. And if we don't hold to those guidelines, we don't get paid. We get complaints against the medical board, and we're outside the norm. But we can't advance medicine without trying to push the envelope. So we need to look at novel treatments. We need the Steve Jobs of medicine to advance what we're doing. Next slide. So with that, we don't want to create harm and everything. So we have these historical measures. When people did research, they looked at range of motion, they looked at imaging, they looked at a lot of things patients could care less about. They didn't look at what patients cared about. How was their job? How was their hobby? And so a lot of these outcomes that were looked at were not really relevant to what the patient valued as benefit. Also, we had varying terms of what treatments were or what outcomes were, so widely varying conclusions and widely differing guidelines. So you have something like PRP. What is PRP? It's platelet-rich plasma. So is that one platelet over baseline, or is that 10 million platelets over baseline? Does it matter? As we heard earlier, is 6 cc's of SuperShot good? Is 12 good? Is 55 better? Or is there a bell-shaped curve where more is not better? And that's hopefully where we're going to get to. And noticeably absent in a lot of these is adverse events, where people are not tracking the adverse events. Next slide. So patient-reported outcomes have come in, and it's nice because they look at a whole range of different components of the patient. They're validated tools, they're completed by the patient, and they give us a much larger picture of the patient's quality of life and impact for medical care. And so some of these measures that we have are either general overall health and wellness, or can be disease specific, such as a hip joint or a particular problem. However, noticeably absent in these even are adverse event trackings. So real world evidence, who cares? Is it any good? Does anybody look at it? Well, over 70% of the time, real world evidence that's been obtained has been impacting the FDA's decisions. So it's not like you do it and you say something and no one really cares. The FDA actually looks at it and takes it as an impactful, component of data. So Databiologics, we're 3 physicians who independently essentially track their own data in a more onerous and labor-intensive way. We got together, we decided to do something. We said, hey, if it works for our colleagues, that'll be great. And we talked to a lot of our colleagues who were nice enough to say, we'll work with the growing pains. We understand it's new. And we had 65 physicians over 2 years putting 3,000 patients into our database with definitely some growing pains. And with that data, we are ending up allowable to get the first OrthoBiologic report published in January 2021 that showed what an overview of our data represented. And so it was nice to know how many patients, how many surveys, what treatments, what areas, and then some rudimentary interpretation of that data, because we just don't have enough yet and for a long enough duration of outcome. But we're moving toward that. So when you think about collecting your own data or using a database, you want to know, is it pertinent to our goals? We think, Data Biologics is perfect for this discussion because we designed it specific to focus on orthobiologics and not get sidetracked with diabetes and a million other things. Our moniker is SUP, simple, user-friendly, practical. So we always try to run through that lens when we make decisions about changing the database. That people may ask, I want 10,000 variables on a patient, but a patient's never going to fill that out. So we'd rather get 10 variables on every patient than trying to do too much. Because there's many people who fail by trying to get too much, and we know of a lot of databases out there that actually just not published anything, despite lots of money, time, and effort. And so our goal is to streamline this so we get actionable data. We want to make it easy for the patient to enter their data, easy for the staff to enter the data, easy for the doctor to look at it, easy to get meaningful data. Make sure we have a high compliance rate. So 10% of patients, 20% of patients responding, not tenable. Is it IRB approved? We're working toward that. Right now, we're looking at de-identified data. You can, of course, put your own data out, but we're working toward various things to try to make this as out in the open as possible. It can be marketable by the physician who's using this in their own office. Cost, whenever you pick something, you want to know, what is the true dollars I'm spending? Not just it cost me $100 and then I spent $5,000 on my staff and they argue with me and I have no compliance with my staff and the patients hate me. You know what? There's more of the cost than just pure monetary. So I'm going to try, if we work well, to link out and show you how easy it is to use this system. So Felix is hopefully going to give me control. Hopefully you can see my screen.
Yes, sir.
Good. So this is, again, a test database that I'll log into that you would have your own email for your company. And from there, this is your sort of dashboard that you would pop up with. You see the treatments that are coming up. These are not real patients. I have a quick overview of how many patients are in your system, sort of a variety of how many patients you have for different body parts. You'd have adverse events that pop up. So you can say, hey, let me go to the adverse event and adjudicate it, whether it's 5 years after that they have a pimple on their nose, or whether it's 1 week later and you treated their knee and they have an infection and went to the hospital. So we want to add a patient. We'll add me, make it nice and simple. Oh, there's a little lag, so hopefully, there we go. Hopefully you guys see the lag is not too bad. And of course, my birthday is not today, but if you want to send me a birthday card, that's fine. And then we'll look at something simple. We'll look at, we talked about, I heard the shoulder earlier mentioned, with some rotator cuff tendinopathy. And then we'll say the treatment date is going to be tomorrow. Patient cell phone number. And that's how they can get the survey sent to them, either by cell phone, which you see these red asterisks, or by their email. And if they don't get it or they think it's spam for some reason, you can always resend it pretty easily. There's a little key, you can just click on it, it'll resend it to them while you're in front of you. Now they're going to get it, and if you're doing this in front of them or your staff is doing it the day before, you can let them know, hey, you're going to get a text right now. You can go to that, start filling out your baseline patient-reported outcome score. And I would go in here and say, it's a tendinosis, it's moderate, or I can check mark this on my pages so that my staff will do this later. I told them it's a subscapularis tendon, and I can add multiple diagnoses in, or I can say it was just a supraspinatus problem. And Dr. Rogers is a great physician, so I'll let him treat me. So if you have 3 physicians in your office, you can pick which one, and you can sort of, I guess, have gentlemen's bets or candy bar bets against who does better on their SuperShot PRPs. And then you can say, oh, I want to do, where is stuff? I can't quite find it. Let me start typing in SuperShot. Boom, there it is. Let me click on that. And then what do I want to use that with? Well, I process my PRP with some M-site product. And my treatment's going to be that. This actually will populate in the real system. What we entered in before was tomorrow's date. So we add that, we're all good. We can add another body part. We can add another site. You'll see over here, as we put the patient in, the circle went around and check mark complete. The diagnosis circled around and check mark complete. My treatment is partially complete. I can either do this now or save it and do it later, have my staff come in after I've actually done the treatment. I can say that I'm treating the supraspinatus. I put 3 cc's in. Did I do another injection site? No, I did not. Did I do it by, my eyes closed in God's guidance, or did I put this under ultrasound guidance or x-ray or both? How much total blood volume did I take to process this? I did 55 cc's into my M site with 5 cc's of anticoagulant, and I had 25 cc's of PPP with my final volume of PRP being 4, and I did not activate it, and I did not dilute it. I can test it if it what happens. Later on, I can go back in and have my staff enter in all my parameters, but they don't have red asterisks, so you're not mandated to do that. And boom, there you set. Back to your home screen, it tells that I'm me, has my contact information, has the diagnosis and details, my treatment date, and then it can tell that, you know what, before I do my treatment, my staff or myself is mandated to look at this and say, you know what, the patient for some reason didn't get their baselines. So before I do the treatment, I have to make sure I resend it or ask the patient to go back in and complete them so I have those desired outcomes. Now, we're going to have, I guess, the screen go back to the presentation. So Felix, if you want to take that back over.
So
There we go. All right, next slide.
So this is then.
So this is just an overview. It's not real data, but this is what you would see if you look at the outcomes. So you can search PRP SuperShot knees, PRP without SuperShot, me as the provider. I can look at the numeric pain scale. I can have 2 graphs along here that compare each other. You can print it out, you can put it on your website, you can mark it because it's your data, so it's reasonable. Next slide. So in summary, real world data using patient-reported outcomes is valuable and generalizable. It impacts daily patient care decisions and may be more clinically relevant, especially to your particular population, than some randomized placebo-controlled trial. The best manner in which we collect these outcomes measures is evolving, and the best ways to engage patient for compliance is evolving, and we're trying to be on the cutting edge of that. The optimal patient response rate is yet to be determined, but depending on the numbers of patients, it may not need to have a 95% outcome rate, and we can try to determine what that would be is valuable and reliable. However, everyone who's doing anything novel outside the standard of care should be collecting their own data, especially people on this call that are thinking about using SuperShot or already doing it. They should be collecting their own data to find out, is that extra cost to the patient valuable? And let the patient decide that in the future. Pooling our data amongst multiple providers will give us lots of information, including which physician does better, which patient group does better, and which treatment is best applicable to that patient group. So there's a lot of work to do. Time to start is yesterday. So again, thank you for everybody who's using the platform. Thank you for the tolerance as we build it and modify it and make it better. And again, thank you for AcCELLerated Biologics and ForeverLabs with the partnership working, tolerance, and look forward to great things together.
Great. Thank you very much, Dr. Bowen. And that was some excellent news. And yeah, the whole importance of data, from us, we want the doctors, you guys, to start writing more papers, ForeverLabs, getting clearances, 510K clearance on effective products in this market space. So we're extremely happy that SuperShot and ForeverLabs is working with Databiologics and getting this program going. So moving forward with the webinar, we were going to get it started with some of the questions in the open forum. One of the first questions that we did have written into our question section: is this accepted in Canada? Can it be sold in Canada and be used in Canada? So, and while that's going on, if anybody wants to raise their hand and ask a question, again, go to the attendee tab and put your little hand up, and I'll call on you next in just an alphabetical order. So we'll just answer that first question about the availability in Canada.
Sure. I'll answer that. So right now, it is, to my knowledge, not available in Canada. I will say this: we are working really closely, Felix, with your team at Databiologics as our national distributor in the United States. If you have people in Canada, you have my permission to start selling it there.
Got it. Okay. All right.
So quick comment from myself for Databiologics. We are actually working with Canada and a new nonprofit called Seaborn to try to get data biologics into Canada. So for any Canadian people on the line, that will be hopefully a option to collect data there in the near future. We just have to make sure we're compliant with all the Canadian laws.
Okay, I'm going to go to Dr. Buford. He has his hand up. Dr. Buford, do you hear me?
Yeah, I can hear you. Can you guys hear me?
Yes, sir. Go ahead with your question.
Okay, great presentation. I wanted to ask a question to Dr. Navity. In trying to compare this to PRP, one of the things we're going to have to do to set kind of a power analysis is trying to figure out what percentage better we think this might be than regular PRP, to know whether we need to have 2 groups of 30 patients each or 2 groups of 100 patients each. And since you got the longest experience, and my question is, do you have a sense of, do you think this may be 10% better? Do you think your patients are, you mentioned 1 month as a potential benchmark for people seem to be doing better?
Yeah, what I've seen so far at the 1 month call is that people are telling me, yeah, week 2, 3, I started noticing a difference. Whereas before, most people hadn't noticed a difference yet, and they really didn't start noticing a difference till like week 4 to 6. The 3 month follow-ups, and then the few patients that I get following up further from that, it just hasn't been a large enough sample size yet for me to comment. But my best guess is it's going to be more like a 25 to 50% difference in outcomes.
Okay. So I think, like a lot of things in orthobiologics, if we start with something where we can all collect a lot of patients fast, like mid-grade NeoA, and then use Jay's platform to collect the data, that would get us to some publications pretty quickly.
Agreed. That is the indication we have selected as the one, KL2.
Yeah, sounds great. Thank you.
All right. Thank you,
Good to
Dr.
see you.
Buford.
Good to talk to you.
Okay. Next up, Dr. Let's see, Dr. Joseph Ruan, Ohio Health. I am see that your hand is up, and I am unmuting you, sir. Dr. Ruan, go ahead with your question.
Felix, did you just call me Joseph?
I might have said Dr. Joseph Ruan. I tried to read correctly. Yes.
Yeah. Hey, everybody. Thanks for this. Jay, first, kind of a comment for you guys. Just I can't thank you enough for this effort in data biologics. I know what you and Chris and Jerry have gone through. I don't know if you're following what's happening with my organization at Ohio Health, but we are absolutely dragging you through the mud, upside down and backwards, through security processes, security challenges, and now legal challenges. But I beg you to hang in there with us. We just sent back a response today from our legal team. They want to redo the service agreement. But what I hope is that when this is all finished, it's a stepping stone for you guys to work with larger organizations like ours. I mean, the purpose for this is to access. I have a multidisciplinary group. I have access to over 25 primary care sports med docs that may jump on board at some point. And Ohio Health has just been brutal. And Luke has been wonderful. And so it's kind of an apology and a plea to hang in there, because I think it'll be important for you to work with large organizations to get over some of these bumps that we're kind of dragging you through right now. So thanks for the effort. Steve and Mark, as we begin to roll this out, I had some curious questions from the staff. It almost felt like they were concerned that we're giving a drug, that this was something that we're putting in the patient's body, and should we have a different consent? Has the question come up at all that you need a different consent for administering this? And then just from a clinical standpoint, does any of the PEG or dextran or any of that enter the patient's body? Are we telling them they're only getting, a better content of their own plasma, or are some of the contents entering their body? And if so, I think we need a consent for that. I'm wondering what your comments are.
Well, Mark, I'll let you talk about the amount of Pegdex and all of that. But Dr. Ruan, first of all, yeah, it's great to hear your voice, and I know you just rolled out SuperShot at your organization, and I appreciate it. We have not had anyone suggest that we need a different consent, but if that's something that your staff feels that they need in order to roll this out, we can certainly go down that path. I'd be happy to entertain that. Mark, I'll let you talk about the PEG and the DEX, et cetera, and what the regulatory consultants came back with. So yeah, but this is me just saying that, Dr. Rowan, I'd be happy to have that conversation if it's needed.
Hey, Dr. Rowan. Yeah, so good to talk to you. And so the PEG and DEX fraction, the determining factor would be, how well you decant it. So it's very low, concentrations to begin with. You're talking about about 1 to 1.2% is the fraction, and then once it's diluted in the PPP, and then you decant off that. We've had it tested by an external lab now on 2 different occasions, and we're doing a third right now where they told us that the PEG and DEX levels are in micrograms per milliliter in there, and we're working towards the 510K right now, and there's a certain level that is below the threshold that it even needs to be reported, and so our initial measurements are right on this borderline of so actually one of them, the PEG is if I remember correctly, is below, and then the DEX was close to this. So I think that what I can say, if you decant it aggressively, you make sure that you've got that PPP off. You're not just kind of letting some the EV fraction plus then some some PPP still in there. If you're getting it, so you just got that EV layer. It's extremely low amounts and probably lower than that. Once we have this, approved device that accompanies with the solution, then I think we'll be able to be able to say even more certainly what the concentration is in there.
Yeah, I think just to pause there, just so you know, Dr. Ron, I know we talked about this, but we are taking the product through the 510K process with the FDA. We're just starting down that journey. Mark, maybe talk a little bit more about Pegdex itself and how it's non-bioactive, et cetera.
Yeah.
Just because, so we are going to have it at a point where it doesn't even, it's not even on the radar whatsoever. We're talking about grains of sand on a beach here, very, very small amounts. But let's say they weren't. Let's say, someone put a good amount, someone messed up and put too much Pegdex, didn't decant the PPP at all and put it in. Talk about what Pegdex is, if you would.
Yeah, so polyethylene glycol and Dextran 500 are both used in injectables. Actually, both the Moderna and Pfizer vaccines use polyethylene glycol in them. That's used often as a stabilizer. Also in polyethylene glycol, is used as a laxative, and you give 30 grams to a child for that purpose. So it's a very inert, it's actually used for the fact that it's kind of inert and non-toxic. Also, Dextran is used in iron Dextran infections, Dextran 500, which is this high weight Dextran, and that's an injectable, and it's used in very high concentrations of Dextran. So they are both used as excipients, and they're used as stabilizers in injectables as these non, active. But we are using them as such. The amount in there, if it's properly decanted, is nigh below the level that is even needed to be reported to even be there. So I'd say be conscientious about that decanting. I'd love to say this is exactly what's in there, but that's going to take, when we do have this approved device, and we can say in this device, and we have that. But it's
one thing I would mention too, Dr. Rowan, as you're talking to your staff, if you're interested in trying to clarify what the process is to them, I think it's really important. This is for everyone. It's really important that everyone knows that this right here is not an injectate, right? This is a sizing mechanism. This works basically as a two-phase aqueous separation. So this is used just to allow for sizing of these small molecules, and in no way is this meant to have any sort of biological activity in patient. This isn't providing any sort of secret sauce to the PRP at all.
No,
as you know, Dr. Juan, but it's probably important that your staff know that too, and I'm sure they do. But if anyone's listening and just joining in, you know, this is not an injectate. It's just used to facilitate the sizing, and that's it. And what's great is one of the stipulations that the FDA allows for in PRP is for rinsing and sizing. And so that's exactly what we're doing here, is we're using this for sizing, as many would use a colloid, et cetera, in different PRP devices that are.
I think that was all well explained, and thank you. And as you said, I kind of knew that. I just want to hear it in a different way. And again, between the legal stuff and this, I just hope I'm helping you guys push some dirt out of the way that you may come across. So thanks for bearing with me.
Absolutely.
Yeah,
absolutely.
always great to talk with you. Thank you.
And Steve, I'll write up. I'll take care of. I don't need any. If there's any sort of separate document or something that we're doing, I'll take care of that on my end. We're good. Thank you.
Cool. Yeah. If you ever need any help, though, we're here for you. Thank you.
Yeah. Thanks.
Great. Thank you very much, Dr. Ruan. I do want to try to see if I see Dr. Robert. Does seem to have logged on here. Dr. Robert, can you hear me now?
Yes.
Great. Welcome. Taking time out of your busy day to join us.
Yeah. It's 4:30 on the West Coast, so I apologize I couldn't attend earlier. So thanks for asking me to come tonight and share my experience with Exosome SuperShot. So I started to use it as soon as it came out this spring, and as Dr. Navity was saying, we don't have a lot of data yet, but I can say that it's making a difference in my practice. What I can notice is that exactly like what you said, it's showing the patient is seeing the benefits faster than a regular PRP. I've been using it in different injuries or indications. So I've been using it in osteoarthritis, but I've been using it also for injuries, let's say partial tear of the supraspinatus tendon and things like that. And I can tell that, I mean, again, we don't have a lot of data, but I can tell that patients have a very good response faster than just the PRP. Just anecdotically, this week I saw a patient for a 2 months follow-up. He had a severe chondral lesion in his knee, and he would have been a good candidate for OrthoBiologics, but they didn't want to go that route with the long rehab and the cost and everything. So he agreed to try with the SuperShot. I saw him at 1 month, and there was still a lot of inflammation and pain. And I was going to convince him maybe that he would need to consider the OrthoBiologic. And I saw him again this week, and to my surprise, 2 months post-injection, his knee is totally dry, pain-free. He's able to go back. He went for a run. He has zero pain. So for me, it's very surprising to get that result with PRP and exosome in an acute chondral lesion in the knee. So I was super pleased with that. The other thing I wanted to share with you is that in October, I attended the TOBI meeting, and I was very surprised that nobody was talking about exosome. So I went to the organizer, and he said, yeah, no, we don't have enough data. We're not aware of much about that. And he was aware of SuperShot, but no more than that. So I said, well, we need to start collecting data and try to be more precise with what are the results with exosomes and to share that with the community. So I went back to AcCELLerated Biologics, and I proposed to do a study, and they kindly accepted. So I have already started and recruited around 15 patients already. So I'm doing a double-blind study with knee osteoarthritis, bilateral knee osteoarthritis. One knee is getting PRP only, and the other knee is getting PRP and exosome. And so the patients have to answer questions, the analog scale, visual analog scale of pain, and the KOOS questionnaire. So at time 0, 1 week, 2 weeks, 4 weeks, 3 months, 6 months. And what I would like to do with this study is, first of all, to see if effectively the PRP with SuperShot knee is responding faster than the PRP only. And also what I would like to see, where are they at 6 months? Is there still some benefits of the SuperShot versus the PRP only? And eventually maybe pursuing this study on a longer than 6 months, depending on the results. So I'm very excited, and patients are interested in the study way more than I thought they would be. And so I've already recruited around 15, and I hope to get more.
It's very exciting.
Thank you, Dr. Robert, for that information. I do have a couple of written questions that I can read to the panel. First one, I guess this is for Dr. Navity. Can you do this with leukocyte-rich PRP? This was from Dr. Buford. It was another question. Or is it only for a leukocyte-poor PRP? What's your answer for that?
Oh, you can use it with leukocyte-rich as well. It comes off the poor plasma, so there's no reason why you can't use it with leukocyte-rich as well.
Okay, great. And another question that was typed in, it's more just of a basic one. As a physician, what is the advantage of isolating the EV layer? What is the message that they want to speak to their patients?
When I'm talking to my patients about utilizing this, it is a added tool or an added weapon, if you want to call it that, that's going to regenerate tissue. So when I'm describing to them how platelets work, that they have signals, that part of those signals that cells communicate from one to the other includes these extracellular vesicles that are otherwise being thrown out. And so by concentrating those and being able to add those back in, we're able to maintain the concentration of your platelets without losing those cell signals or those other extracellular vesicles.
We have open forum left. We have a few more minutes left. I don't see any more hands raised or questions to be answered to the panel. So if I don't see anything here right now, I just want to let everybody know that is on this webinar. Obviously, it is being recorded. We will be able to post this on our website and our YouTube channel when everything is processed, so you can share it with friends, other physicians, go back and review what we've discussed today. Also on the screen, that if you are registered for this webinar and you would like to try some more SuperShot, if you've already done some or try one or two for the first time, you can reach out directly to us at AcCELLerated Biologics. You can reach out directly to your individual distributor. But we want to make it clear that we're doing this as efficiently as we can, getting just the right information to all of you physicians so that you feel confident with the product, confident with the science, and we've kind of built a nice group here where data is being collected. It's an easy way for everyone here to make a real positive impact in regenerative medicine, PRP, SuperShot, and on down the line. So if I don't have any more questions, which I don't see right now, I'm going to end this right now. But if there's any last comments from any of the presenters, I'd like for you guys to kind of have the last words.
Yeah, Felix, can I just, I guess, ask a participant question, we'll call it?
Sure.
And probably aimed more at Steven. You know, words matter, legality, the 36-month moratorium is over, the FDA, FTC are looking at things. And maybe in Mark's comments, I mean, there's a difference between science and regulatory speech. And I've heard tonight a few different things, and I'm sensitive to it only because I think this field is extremely important, valuable, and if we make sure we police ourselves and we advance the science, it'll be around for our patients. But, you know, we talk about EV, these extracellular vesicles, and we talk about exosomes. And exosomes have had sort of a negative connotation on the regulatory side for 36 months, being allogenic mostly. And is there anything that you've heard regarding your product about how it should be termed, things that you're limited in saying, whether it's advertisement from the FDA or what you talk to your distributors about, or what we as physicians should be careful about?
Yeah, the truth is, when we first started building the product, we did refer to exosomes quite a bit more than we do now, and the reason is because of the crackdown on what, as you correctly stated, the allogeneic exosome products that were in the market and other allogeneic products. As I said at the outset, ForeverLabs we're interested in autologous therapies and leveraging autologous treatments. It's what we started with, banking young adult stem cells, et cetera. But to get right to the truth of it, we switched over to saying extracellular vesicles for two reasons. One, it helps us not get confused with allogeneic products by calling them extracellular vesicles. And number two, it's accurate. What we are concentrating in this product isn't just exosomes. There's a whole slew of these extracellular vesicles in there. I think exosomes from a marketing standpoint is actually probably more, I'm the CEO, I'm not a scientist. I'll put on my marketer hat. Exosomes probably would be easier for us to go out there and market because consumers have heard of them, they're more aware of them. But the truth is what we're concentrating are extracellular vesicles. There are exosomes in there. I think it makes sense for us right now to be calling them extracellular vesicles because number one, it's accurate, and number two, it helps us avoid being lumped in with these allo products that have been in the market prior to us coming in. The truth is when we first launched, we were so excited. We were like, this ruling coming from the FDA is actually going to be wind in our sails because of we're the only person left standing. We have an autologous product that we can offer in the marketplace. But the truth is it made us have to reevaluate how we bring it to market, how we talk about it. We check all of the right boxes. We've had FDA regulatory consultants take a look at the product, and we know that we are autologous, homologous, point of care. We fall under the regulations for PRP that already exist. So we check all the right boxes. The question is, how do we mention it? And I think that's really kind of gets to the crux of what you're asking. We talk about extracellular vesicles, and you can say that in the extracellular vesicles, there are exosomes. I mean, it's all accurate. Mark, anything you would add to that?
Yeah. So I hope you picked up. I'm kind of, I'm a scientist. I'm a bit conservative. I mean, I have an allergy to unqualified statements. Like I have a difficult time making a statement without, adding the qualifier because I want to be, truthful. What excited me, why we really, the truth is that when we were doing the mesenchymal stem cell, treatments, we were developing those, we just started to find that, oh my gosh, these EVs that they're producing, which exosomes was like a large portion of it, are such a large part of the therapeutic effect that we started to say, well, maybe we don't even need the cells. Maybe we just need the EVs that they're producing. And then there was kind of a burgeoning of this, research into extracellular, vesicle therapies like EV exosome therapies and whatnot. And there's been an explosion in that space. And so when I saw that the plasma we weren't concentrating this, it was just, that's what really, made me say, wait a minute, this is an opportunity, not an opportunity just like to make a whole bunch of money. That wasn't like the interest. The real opportunity is like, oh my gosh, this PRP can be improved. Like this is being used, and look at this being thrown away. In my heart of hearts, I really appreciate what Databiologics is doing. Like every patient that isn't, every time you treat a patient and you're collecting data, you're doing a disservice to a future patient because that's an opportunity to learn. And it's just that's why we want to apply that to what we're making available. Like, hey, you can concentrate this EV fraction. It's already in your PRP. It's there. It's probably actually helping. It's probably, but and so yeah, the language around it. I think Dr. Navity has a good way to talk about it. Maybe you'd say, it's a 10-year whiskey is good. A 20-year whiskey is even better. This is a 20-year whiskey. It's the same ingredients. It's the same stuff, but yeah, it is, it's the same stuff you've been working with, just concentrating one of the fractions that you haven't been concentrating.
Yeah, one thing too is seem to respond, patients seem to respond to analogy quite a bit. And when Mark was talking earlier about how in the lab they had to use previously a washing machine size centrifuge to run overnight they gather what we're now in 1 minute able to add to the PRP. I think that if I was a patient, I'd be like, wow, that's really, first of all, really fascinating. And then I would ask, what do they do? And to Tom's point, well, they're communication packets that go out into the body and request other cells to areas of injury to help, accelerate healing. I think that's how I would describe the product.
Okay, gentlemen, ladies, everyone that attended, I appreciate your time. It's holiday week. Everybody get ready for a great Thanksgiving next week. And we will be able to answer any more questions that weren't able to be asked tonight. Please give us a call, give us an email. We will be more than happy to reach out to anybody on the panel. Everyone's email was listed prior to tonight's webinar. So anytime you want to communicate further, we'll be more than happy to help. And thanks again for everybody for joining us. So have a good night, everyone. Thank you very much.
Good night. Thank you.